If you think about DNA as source code, this is debugging the source code, right? One letter at a time. It's not chopping out big fragments. It's more like the eraser on a pencil, where you can go in and, in a very targeted fashion, make these edits to the DNA code.
Hi everyone, I'm Nikki, and thanks so much for tuning in to this episode on gene editing. I recently caught up with Dr. Tiffany Vora, Vice Chair of Medicine and Digital Biology at Singularity University, to get her thoughts on its future and whether it might be offered as an employee benefit by the workplace. I think it's something that could really offer huge advancements for our species and also our planet. I came across something that I never knew before, where you shared that every human being is identical to every other human being, with 99.9% of our DNA actually being identical, which means 0.1% of us is actually what makes us unique. How does gene editing actually manipulate this?
Because all life on Earth is related, all life runs on the same operating system using the same source code, which is DNA. Now, DNA is made up of these 4 chemical building blocks that for short we call A, C, T, and G, and it's a physical molecule. What's amazing about gene editing is that gene editing is a technology, a biological technology that can go in and read out the order of those A, C's, T's, and G's, find a particular sequence that it's interested in, and then change that sequence. So interestingly, we actually had gene editing for a while. There were previous technologies and approaches like zinc fingers. But the whole point with no matter which flavor of gene editing you're talking about, it's not something that humans invented. These are systems that evolved over time through nature to do particular biological tasks. And the cool thing is humans recognized that you could grab that biology and then use it for something else. So let's look at the CRISPR-Cas system, and I'll just tell you quickly how that works. So the CRISPR-Cas system was originally the bacterial immune system. So this is the way that bacteria are able to recognize viruses that have infected them before and then be ready to attack that viral code when it comes into the bacterium. And the bacteria does this by chopping up the invading viral genome with a pair of what's essentially molecular scissors. And you can think of Cas, which is a very small protein, as being like a molecular hand. Now, you're probably saying, well, wait a minute, I don't want something in my body that's running around and cutting up DNA. And for sure you don't, and the bacteria didn't want that either. And that's why Cas evolved to take what's called a guide sequence, where you tell the molecular hand and the molecular scissors, I am interested in this particular sequence of A, C, T, and G. And that particular strand of DNA goes into the molecular hand and guides the scissors to that exact place on the genome. And when there's a match between the genome and the guide, that's when Cas can act as a pair of molecular scissors. So you can think of it like the CTRL+F on your computer. Instantly, the cell wants to repair that DNA. And normally what it does is it looks at your other chromosome to see what sequence is supposed to be there where there's a hole right now, and then it copies that. So what the human scientist does is it tricks that system, and you flood the cell with a bunch of DNA sequences that contain the edit that you want to make. So what happens is the cell's repair system gets tricked, and instead of copying the thing that you were getting rid of, it copies the template, the new edit that you're putting in, and then very nicely glues that new sequence into the genome. So that's how gene editing works, and it's really fascinating. If you think about DNA as source code, this is debugging the source code, right? One letter at a time. It's not chopping out big fragments or swapping big fragments. It's more like the eraser on a pencil, where you can go in and, in a very targeted fashion, make these edits to the DNA code.
I love that analogy of debugging. Are there any particular use cases currently that are being used to debug and potentially extend human life?
Oh my goodness, there are so many use cases. So you mentioned human life. Let me first point out that because all life on Earth runs on the same operating system, if you can do this to human DNA, you can do it to basically any other kind of DNA. So for example, in agriculture, we are seeing lots and lots of use cases for this technology to come up with new plant breeds, to find ways to fight pests, to fight drought, to fight salination, to fight global warming. All of this is happening in agriculture in really exciting ways for plants and animals. And in fact, what we think is going to happen is a lot of the first approvals for these processes are going to come from agriculture, because the regulatory requirements for agriculture are different than the regulatory requirements for human medicine. There are several really exciting use cases that are happening right now for human life. So this is not science fiction. This is happening today. Gene editing comes in two flavors. The first flavor is called somatic, soma meaning body. And what that means is when you make the edit stays in the body cell basically where you've put it. So think about a skin cancer, for example. If I have a tumor on my skin, on my arm, and I use CRISPR to change the tumor DNA into being healthy DNA, then I wouldn't pass that on to my children, because my skin doesn't get passed on to my baby. Now, in contrast, the second flavor of gene editing is called germline. And what that means is the change that I make gets passed on to the next generation. So if I change my eggs, if I change my partner's sperm, or if I edit an embryo before it grows into a baby, then not only will that child have the edits that they got as an embryo, but their children and their children and their children will have it too. So what we're seeing right now today for use cases are somatic use cases, where only the body is being affected and it doesn't get passed down. Just the other day, there was some exciting news from studies using gene editing to treat hereditary blindness. We're also seeing clinical trials going on today using this technique for cancer immunotherapy and also for inherited blood diseases, so sickle cell anemia, beta thalassemia. Some folks are working on Duchenne's muscular dystrophy. And I recently learned about a company that's using CRISPR to fight persistent urinary tract infections in the bladder, right? Because the bacteria, they get in there and then you can't get them out. And so CRISPR is an exciting way to do that. So not science fiction, happening today. There were already companies trying to use gene editing to cure HIV. And you do that by changing the shape of the molecular door that HIV uses to get into T cells. There's so much innovation going on in this space right now. As we solve the easy problems, the low-hanging fruit, things like blood diseases are great test cases for this technology, because you can take the blood out of the person, work on the blood in the laboratory, and then put the blood back in the person. That's much easier than trying to deliver these gene editing drugs, reagents, to parts of the body that are much harder to access. So the more success we get, I think the more people will come to trust the procedure. I think the more scientists and companies will learn what works and what doesn't. And at the same time, we're seeing exponential advances in our understanding of human biology and human DNA and how it's linked to disease. The more we learn, the better we can target these types of procedures to really positively benefit as many people as possible around the world.
You touched on something actually quite personal, which was all around children. Many women go through fertility issues, myself included. Do you see this technology surpassing IVF treatment and actually offering women a better solution?
So there's two interesting issues in what you've brought up, and I'll take the second one first. So I think of gene editing as an add-on to IVF. If you can take something out of the body and make the change, that's much easier than trying to do the change in the body. We cannot point to one place on the DNA and say this place on the DNA causes that problem. Now, these are called Mendelian diseases when you can do that. And we know of a few thousand places on the genome that cause a few hundred Mendelian diseases, where we can point and say, this is the problem, this is the disease. So those use cases that I just told you about, Duchenne's muscular dystrophy, beta thalassemia, sickle cell anemia, those are all this type of disease where we can point to one place and say that that's the problem. In contrast, most of human disease is, first of all, a mix between what goes on in your environment and a mix of what you've got in your DNA. And on top of that, the DNA part isn't just one place on the DNA, but many, 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 many places on the DNA, each of which has a small effect. And when you add them together, that's when you see the bigger effect. And so complex situations like infertility are likely a result of many, 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 many places on the DNA. Now, the good news is as we get better and better at looking at those many places, identifying them, and really quantifying how much does each place have an effect, and what are you doing in your daily life? What are you exposed to? What were your parents exposed to? All of that information can be integrated into a personalized treatment strategy, but not today. I think that's down the line, but it's possible. So I think for these more complex issues, it's not going to be a silver bullet solution from gene editing. And honestly, Nikki, I think I'm okay with that, because reducing it down to a single place on the DNA wipes out that personalization of a person's experience. And so I kind of feel like this is a yes and rather than an or.
There's many workplaces today that offer private healthcare benefits to their employees to ensure that they are remaining healthy, happy, so they can do their most fulfilling work. Do you see in the future that the workplace may give employees at least the choice to be able to use gene editing as a preventative treatment?
Yes, I do think that is possible. I'm of two minds about that. I can see both the pluses and the minuses. For the pluses of having your workplace provide support for this type of thing, I think that could go a long way towards closing accessibility gaps. I'm not really interested in a future where only rich people can make sure their kids don't have cancer. I think everybody should have access to this technology. And so the idea of going through the workplace as part of this transition to making it broadly accessible, that's exciting to me. I think that's interesting. Now, the parts where I get a bit worried are places where I have to ask questions like, how much would I want my employer actually knowing about me? Do I want my employer knowing something either about my entire DNA sequence or about just specific places, like let's just say oncogenes, genes that we know are related to cancer progressions, genes like P53 and RAS, for example. Now, in the United States at least, and in several other countries, there are laws specifically that say employers and insurance companies cannot discriminate against people based on their DNA sequences. You can't be fired for carrying a cancer gene. But now if we're saying the workplace is in a position to be offering a treatment for that DNA, well, if you were going to apply for that, you would have to give them the information, right? So then what happens from there? I'm not saying these are unsolvable problems, but I'm saying they are complicated and they have some interesting knock-on effects. Thinking about, what do you owe your employer? If you are an employer and you are trying to extend the health span of your employees, well, if I just extended your health span by 20 years, I paid for that as your employer, and now you quit on me and go work for someone else, what has happened to my investment? That's kind of a weird question, right? And the more we mesh the fundamentals of human health and human biology and maybe even human identity with the workplace, the more I think we need to have these uncomfortable questions in a safe space with a variety of stakeholders. I mean, Nikki, for me, it's all about choice. And that is where I would want to see the future go, that you would have the choice to access these procedures rather than a mandate to do so, either from your government or from your employer.
I would totally agree with you. I think choice is always a really positive thing, especially as digital technologies continue to change the way in which things are done or think about how they may be approached in the future. One of those choices may be that you are one of those people who would look at any pain and adversity that you experience in your life as your destiny. If you were to edit something that you were meant to go through, and you were meant to go through that pain because it was going to help lead you to another part of your life journey, would it change that life path in a way that it was never intended for? What are your thoughts on that?
DNA is not destiny. Your health, your biology is an interplay between your DNA, your environment, and also the behaviors that you enact. So if you have this idea that being able to edit your DNA means that you don't have to live your life and engage in behaviors that support health, then there's no point. You hear stories about folks who get stomach reduction surgery, but then don't change their habits, and so it balances out. You don't actually see any results. And so the idea that fixing your DNA is the only thing that you could do to fix the problem. It might be necessary, but not sufficient to do that. And I remember in our initial chat, I told you a story about a talk that I had given once for a group of students in Canada. And we were talking about gene editing, and one of the students said to me, well, this is fantastic. We should be using this to erase depression. And I said, well, first of all, depression is complicated. It doesn't live on one place on the genome. But I said, but also remember, without depression, there'd be no jazz. There'd be no poetry. We wouldn't have the blues, which is one of my favorite musical forms. And so I think it's easy to have this idea of what good looks like that kind of elides the variety of human experience that has made our cultures and our lives so rich and so beautiful. One of the things that I think really defines people is this capacity to take suffering and tribulation and adversity and transform it into something new. I'm not saying that we shouldn't use gene editing to cure human disease, to prevent human disease. I do believe that we should do that. But I think we need to ask very carefully, what are we doing this for? What are the outcomes that we want? Who are the people that we want to be as individuals and as a species? And what choices am I willing to make for my generations who come after me? Those are really exciting, hard questions. And I'd like to invite all of our listeners to have a conversation like this over your dinner table tonight with your family, because everyone is going to come at this from a different angle, which is why I land back at choice.
Thank you so much, Tiffany. This has been a really thought-provoking conversation. I think it's definitely going to be a conversation we have at my dinner table. If people want to stay in touch with you or stay close to what you are doing, how can they go about doing that?
You can come find me on LinkedIn. Make sure you mention that you heard this podcast. You can follow me on Twitter and on Instagram. And there's just lots of really great stuff going on out there. I like to share the opportunities, places that I'm speaking, workshops, programs that I'm participating in, and it would be great to see some of you there.
Thank you so much, Tiffany.
Thank you.